So my name's Noah Falgren. I'm the Director of Data Science at the Donald Danforth Plant Science Center. Drove up from St. Louis yesterday. Um, I made my talk different than I think I normally would. I figured since I was first and that we would have a mixed audience of HPC folks and plant phenotyping folks that I would talk about what plant phenotyping is. So if you already know what that is, then sorry. Um, my group data science is a core facility at Danforth Center. So I do work at the intersection of HPC um, and general like research kind of computing, but my group focuses particularly on plant phenotyping problems along with others. So my team is on the left, a great group of folks, including data scientists. We have a data science trainer, which is great. So we do internal workshops a lot. Statisticians. We work a lot with Malia Gann's lab and Katie Murphy's group, which is the phenotyping core facility at the Danforth Center. And both Malia and Katie are at this conference. So I recommend chatting with them. And then our funding sources are on the your right. So what is plant phenotyping? I like to think of it as um, the measurement of physical, physiological, morphological, or other features of plants. And there's not a technology implication in that. You could be doing that by hand. You could do it a lot of different ways. So that is plant phenotyping. But typically, when we talk about plant phenotyping, what we're talking about is doing it through a kind of phenomics approach, so like a high throughput approach. Um, and a nice parallel I like to think about is genomics. So for the last couple decades, DNA sequencing technologies have made it much, much faster and much, much cheaper to measure or to read DNA sequences. And that led to an explosion of genomics techniques. So anything that can be read out by sequencing DNA can be assayed really easily with these approaches. And so then the question is, can we do the same thing with phenotyping or phenomics? It's harder because there's not just a single kind of, we can't just read a single kind of molecule and understand phenotype. We have to measure lots of different things. So that's a challenge. And phenotype is this integration between genotype, time, and environment. So as I mentioned, genotype is relatively easy and cheap to assay, but environment and measuring things over time are more challenging. So a general approach that I'll talk about today is using image or image-like things to measure phenotype. And the goal is to make phenotyping more quantitative. We want to increase the amount of space and the amount of time that we can do measurements. Another aspect of phenotyping with things like images and sensors is that instead of having to destroy the plant to measure it, we can measure non-destructively. Um, and then, of course, if we can be successful at those things, we can reduce cost per measurement in theory. So because phenotyping is a complex problem that involves both plant biologists and engineering and computer science and many other fields. It's a very multidisciplinary field, and that was one of the reasons that we've built a community called the North American Plant Phenotyping Network, which is a sister organization to the IPPN, which is hosting this conference, to bring together these different communities to build that multidisciplinary community so we're all working together. And so we have an annual conference to do that work. usually in February. Another complicated thing about phenotyping is it is done at vastly different scales. So microscopy actually has probably been way ahead of what we think of as plant phenotyping, but we can do imaging at cellular scale, plant or organ scale, um, controlled environment scale, all the way up to field or even like terrestrial scale, using microscopes all the way to satellites. Um, so while the imaging is kind of the common denominator, the computational techniques in that range of scales is pretty diverse. At the Danforth Center, we do work at these different scales. We have a core facility called Advanced Bioimaging Laboratory, which just does a lot of cool microscopy things. Phenotyping core facility, a field research site where we partner with the Taylor Geospatial Institute to do aerial and satellite imaging. So this kind of gives us that test bed to work on these approaches at these different scales. If we can be successful at developing these technologies, we can impact, these are my views of where we can make impact in sort of the chain of things that are required to address grand challenges in plant and agricultural sciences. So foundational research, crop breeding, precision agriculture, horticultural automation, and post-harvest evaluation. And these required somewhat different approaches, but some of the underlying techniques are kind of in common across these different areas. Okay, so my team, as I mentioned, works kind of at the intersection of computing and the application of computing. So we'll talk about phenotyping. So we run a HPC, which is now on Amazon Web Services, and we do software development in the plant phenotyping space. So I'll talk about PlantCV and a couple of related projects today. These are some of the platforms that we work with at the Danforth Center, though we do work with other people's data. So we have the Bellwether Phenotyping Facility, which is a little over 10 years old now. If you have a chance to take a tour of the phenotyping greenhouse here, it's a similar system in that it's built by the same company, Lemnitech. Ours is a large growth chamber with an imaging loop. So the plants are moved through the imaging loop once per day to do color and a special type of grayscale imaging called near infrared. Yeah, so they're automatically watered, very controlled environment, very regularized imaging. Other things we do are like low-cost phenotyping with Raspberry Pi cameras. Malia Gian's lab has a hyperspectral camera on a robotic arm. We do X-ray computed tomography and traditional aerial imaging at our field site with uncrewed aerial vehicles and the like. So to leverage that data, about over 10 years ago now, we built an open source Python package called Plant Computer Vision, or PlantCV, and it uses a lot of standard Python libraries for computer vision and data science, brought together in one package with one interface so that you don't have to, like say a biologist using our package doesn't have to learn all the underlying tools, and one, what I think of as nice set of documentation to bring it all together. I think we recently interviewed people for a project we had, and somebody told me that OpenCV effectively has no documentation, for instance. No offense to OpenCV. We have a team that builds PlantCV, maintains and develops it, but we've also worked with over 50 contributors from around the world who've happened upon the project and volunteered to contribute to it. And we're at about 140 total papers or graduate student dissertations that have used PlantCV in the research. So we do treat it as a professional software development project, and I think this is important for maintainability, sustainability of the project. So instead of people kind of, because we have a multi-contributor team, we need to manage contributions so that we're not stepping on each other's toes. So a person would typically start with their local copy of PlantCV, make changes, submit those to us as a pull request on GitHub, and then this kicks off an automated chain of checks. So we do unit testing, all of the old code plus the new code is tested. And then there's automated code quality analysis and code coverage, meaning like what code is actually tested. And once those checks are done, we have a human maintainer that reviews the contribution along with the results of those tests. That often leads to around like rounds of revision. It's just like submitting a scientific paper, but there's no reviewer too. And once that cycle's done, that gets merged into PlantCV, becomes a new version, new documentation is built, all previous versions of documentation are maintained, and periodically we release a named version, which is posted to the Python package index, Conda Forge, and archived at Zenodo. Then a person who wants to just use PlantCV and is not a developer can just install the package from one of those two sources. More recently, we've kind of been building out a larger ecosystem of PlantCV because we've had needs to move beyond just controlled environment phenotyping. So we have tools we've developed for geospatial, meaning like mostly tools to work with whole field imaging from like uncrewed aerial vehicles, but some satellite work, and a package for image annotation where we can use PlantCV to semi-automate the process of annotation for downstream applications with machine learning. And just released our package called PCVR, which is a statistical data visualization and quality control tool that takes PlantCV outputs and does the hard work of converting numerical measurements into like biologically meaningful results. Okay, so just a couple examples of what this looks like at the different scales I mentioned. So here's at cellular scale, we have an image of pollen grains on your right. So the goal would be we want to like say count or phenotype pollen, the individual pollen, and whether they're germinated or ungerminated. So on the left side, we can auto-detect the pollen pretty easily. Some of them are touching, so we can't tell them apart. We can't tell whether they're germinated or ungerminated. We can just detect them. With the annotation tools, though, we can load those pre-detected pollen into the annotation tools, so we have all the red X marks on the left preloaded. Then the user can correct those annotations by adding or subtracting markers. Plus, on the right side, they can mark as a different class the germinated. So then, instead of having to manually annotate all of those by hand, you only have to annotate a few dozen. So it makes it a lot faster. So it's not completely automated, but it's also not completely hand labeling. And then sort of the bread and butter of PlantCV is this controlled environment plant kind of scale example. This is just an example from our Raspberry Pi imaging system where we have time lapse over a tray of plants. These are Soteria plants. We can segment the individual plants from the background and then measure those at each time point. And then at the field scale, some of the things we've been working on is abilities to identify experimental plot units, particularly in cases where there's definitely a lot of tools for this. We have cases where we don't have super uniform plot layouts, and so we have some needs to be able to make, I guess what I'd call wonky field shape layouts. So once we can do that, then we can kind of use existing PlantCV tools to take those and extract things like vegetative indices. Okay, so very briefly, PlantCV is a modular toolkit, and so it has tools like the ability to convert between color spaces, do segmentation with thresholding or other methods. You often need to do some type of filtering to clean up those segmentations. We have spatial filtering, and then once you can get down to plants labeled and the background removed, you can do analysis like shape and size, color, spectroscopy, depending on the kind of data you have, and all sorts of things like that. So the way this is done then is a user takes the modules in PlantCV and builds a workflow. So the workflow is customized to the kind of work that you need to do and the kind of data you have. So the typical workflow would look like this, where you're going to import data into PlantCV. You could optionally do some visualization steps to help you figure out how to build a workflow. And then you're typically going to do some type of conversion or transformation on that data, segmentation, filtering, spatial filtering, say to like, if you have a color card in your picture, you might need to like remove that from the analysis part, and then analysis of whatever you want to measure, shape, size, color, etc. And then on the right side is just an example of one of these workflow steps where you can see what all the intermediate kind of transformations on the input images are doing. And we do this in Jupyter Notebooks. So I'm just going to show a video of launching one of our interactive tutorials in the Binder environment. So this is going to open a Jupyter Lab instance in the Binder cloud with a preloaded notebook and example data. But a typical user would develop a workflow by coding in Jupyter. So each cell would be a logical step of a PlantCV workflow so that you can visualize the input image and the effect of the step as the output image, and then make adjustments on the fly as you need to. So the ability to do that like kind of asynchronous step work is useful, so you don't have to run an entire script every time you want to change one parameter. So once you build a workflow, ideally you're in a situation where if you have a fairly like a standardized set of images in some form or another, you would then want to take that workflow and do batch processing on your data set. So that's mostly what we work on. So to do batch processing on HPC or not, we have a tool that can like process data sets. So it needs to extract metadata from that data set. We typically do that like because of the kinds of systems we work with and other people work with. That's typically through file name metadata. Most people we found tend to just name their files with strings of metadata information. So we extract that metadata from the data set and then create basically a task list, which is 1 to hundreds of thousands of tasks, and then batch process that on HPC And so that we don't have to program an interface to everyone's unique HPC system, we use Dask Job Queue as the workflow engine. So it knows how to talk to HT Condor and Slurm and PBS and whatever your favorite scheduling system is. And yeah, so those, it creates, yeah, question. Yeah, the question was,
Thank you so much. So the question was like for HTCondor, I work with WasteG or Wastepool. We don't have a shared file system. So some of the workflow management tools, they don't work pretty well with us. So for your case, like for Plant CV, does it work with HTCondor without a shared file system, or does it need to have a shared file system?
I would say out of the box, the way we use it is with a shared file system. I think though, well, yeah, no, I'd say like right now you have to have a shared file system. It would take some extra work to, like theoretically, I think you could build in like a file transfer piece into the PlantCV workflow. Since the PlantCV workflow at this stage is just a Python script, so you could add in information that tells, like you could program in how to retrieve the data from a remote server. That would be possible, but not like an automatic feature of PlantCV. Yeah. Right now it's expecting that every worker has the same shared file system.
And as I already interrupted, you can ask one more
Yeah,
question.
absolutely.
So uh, I have zero background with biology or plant side.
No worries.
The type of computing that you, or the image that you were showing before, uh, just it looked to me that you can do a lot of processing of those things without requiring much of computing, in terms of you can do parallel processing and you don't need to have a lot of RAM or CPUs or cores to load that. So
It's a
uh,
That's a good question. I think my next slide or the slide after kind of deals with, yeah, like that the computing needs depending on the kind of data you have.
okay.
And some stuff that we're not doing in PlantCV, but that I like I'm just gonna post up here for discussion about yeah, like the different data have kind of different computational needs, and I think that's an interesting thing to talk about for sure.
Okay. And
Yeah.
just to follow up with the first question, so say a researcher from plant side, they come to us, how easy or how easy or difficult it is for them to, like, if they want to implement the non-shared file system or have a different
Ah,
source for a file transfer mechanism?
um, I I think it depends on what it probably just depends on like what needs to be done. Like in terms of like if you program it yourself into a PlantCV workflow, I think it just depends on the complexity of like the how the fetching is done itself. But if the other way to look at it would be the PlantCV team is happy to support use cases that we haven't already thought about. Usually we just need to be pointed in the direction of like what's the problem that needs to be solved, and we're happy to tackle those things. Um, but yeah, both are possible. Users can kind of work around our limitations, or they can work with us to just get rid of that limitation. Yeah, we just need the use case.
Yeah, thank you so much.
Yeah. One last thing that's nice about Dask is it doesn't require HPC. We have it set up so that if you just have like a single machine with like if your desktop is powerful enough for what you need to do, it will also do the scheduling like on your local system. In computational terms, that means you can either use their like local cluster client or you can use one of these other infrastructure clients. Yes, my next slide. So this is made up, but But generally speaking, like RGB data or grayscale data are small per file, but because of that, they're also easy to capture. So those data sets tend to be much larger, say like in the upwards of hundreds of thousands, maybe even millions of images. And so those really fit really nicely into the batch processing use case. Again, that could be just on a single big machine or distributed kind of thing. Computational power per task is very low. Single CPU, typically low memory requirement. As you move toward things like the uncrewed aerial vehicle, which isn't necessarily, they could be relatively small, but they also could be huge. It depends on the kind of like field scale and flight altitude that you're doing. So they could be very high resolution and pretty large. Or hyperspectral, which are very, very large images. But because they're very large and they're actually pretty expensive to collect and time consuming, you tend to have many fewer of them. So batch processing becomes somewhat less of an issue, but then you need more computational resource per image, like high amounts of memory and things like that. Yeah, sure. So I'd say on my image size axis, on the left side, we're typically talking about a couple megabytes, like maybe 5, 10 megabytes per image. On the right side, hyperspectral could be in the tens to hundreds of gigabytes. If in the UAV context with hyperspectral, theoretically upwards of terabytes per image.
Have like all of all of sorry, I can't. And from a researcher's point of view, do they need to have the full image at once in a single machine to compute, or they can segment and like filter out and just
You're just predicting everything I'm gonna say. So these are the challenges that I see as like maybe my own personal challenges, not necessarily all community. Those some of these are community challenges, some of these are our personal challenges. So exactly with the data sets that are very large per image, where batch processing doesn't make as much sense. I do think we could do better with like parallelizing operations on those images themselves. Like we don't need to do that on the RGB dataset side. But yeah, like with hyperspectral or other large data structures, data chunking could be really useful. So Dask does that. We could do that natively because all of our data structures are just Python NumPy arrays, and that's what Dask is really good at. Or there's also multi-threading, our GPU computing are also kinds of things that could help us accelerate computing on like those really large data matrices, for sure. Yeah. So I think like I've tended to stay away from that in the PlantCV context because we mostly work with the smaller data and you things start to get a little bit dangerous, I guess, when you allow a user to do batch processing with multi-threading at like really large scale, just because people can kind of schedule pretty, like, if you're doing like hundreds of workers times like tens of CPU cores, it gets a little messy. Yeah. So other challenges would be things like typical software kinds of things like reproducibility. Um, how does a user keep track of what version they were running, what version of all the dependencies were in their environment, um, data set descriptions, that gets into things like the interoperability, like data standards. So things that we are always keeping in mind are things like REMBI and MyAppy, which is minimum information about a plant phenotyping experiment, and BRAPI. Um, and then data sharing can be challenging. So for small data sets, we can share with Zenodo if it's up to 50 gigabytes per data set. Figshare and Dryad start to have cost if you go pretty big. And then for larger data sets, things that we are looking at are eDAL, which was developed by IPK and Bioimage Archive at European Bioinformatics Institute. Since there's, unlike genomics data, there's not a national repository for this kind of data. With that, just thank my team again, and yeah, happy to answer any other questions.